So there's my thylakoid disc, right? That's the membrane that you guys were just dealing with. Inside the membrane, you have lumen, and that's where you have the proton gradient building up so it can flow back out. So that's where you have the water that you're splitting. On the outside of the membrane, so in this kind of space here, anywhere outside of the thylakoids is the stroma. Here's the word here. And the stroma is the jelly where I'm doing this next set of Calvin cycle stuff, where I'm grabbing the carbon dioxide. So carbon dioxide is floating around in this area, and so is the rubip, and I'm going to crunch them together. So this is actually where I'm making my sugars as well. Once I make those sugars, they can just leave. They just head out, and they go to, like out here is cytoplasm, right? Because this is my chloroplast, so out here is my cytoplasm. So when you take another look at our membrane, okay, so there's my NADP. Where am I? I'm in the stroma, right? And here's my, let me scroll down. Here's my ADP. Where am I? I'm in the stroma. So they remain outside. There's my ATP outside. My NADPH is outside. Everybody stays outside in the stroma. So they're created on the outside edge. So this is the outside edge, right, of my thylakoid membrane. And they started in the stroma. They remain in the stroma. The final product is ATP of this stage. Now I'm going to use them in the Calvin cycle, which is what we're now talking about. So I did all of this monkey business here in order to make NADPH and ATP because that's energy captured, again, stimulated by the light, but energy captured that I can then use for exactly what I'm about to do now with the carbon dioxide. What we have here is the light reactions. This is what we've just studied this morning. We've gone through this conversation. The goal was to produce ATP and NADPH. We had one way to produce NADPH. That's if the electrons were lost and I reduced NADP to NADPH, and I had to replace them ultimately from water. That's what made it non-cyclic, that the electrons didn't replace themselves. But I have two ways to make ATP, or at least two processes complete. I can use that same non-cyclic, but I can also use cyclic and have the electrons replace themselves. The key would be that as long as I'm activating that proton pump that's part of the B6F, I can always be pumping protons. And as long as I'm pumping protons and making a proton gradient, I can always let them flow back through ATP synthase. As I'm saying it out loud, can you see the membrane in your head? Okay. Then you are at a comfortable place. If you can't yet, keep working with those membranes and keep talking about it, working it out for yourself, saying it out loud so that you can get there. Okay, so what are we going to use ATP for? Well, it is our energy source that we are using in this very next stage, and the same thing with the NADPH. It is a reduced carrier, reduced because it's carrying electrons. So it's an electron carrier, which means it's going to be a reducing agent, an agent of reduction, right? Reducing agent means that it's going to be the agent that causes something else to be reduced. It has been reduced itself. When it gives away its electrons, it reduces somebody else, therefore agent of reduction. Hopefully that helps. So NADPH is the reduced carrier. It is a reducing agent. It will reduce something else. And what is it reducing it with? A high energy electron. This is where we have captured that light energy. We've excited our electron. It's very, very excited, very high energy. And we're going to use this energy now to help me form the sugar that I want to do. So ATP, NADPH produced in the light reaction will now be used in the light-independent reaction. The light-independent reaction happens in 3 stages. We fix the carbon, which means attaching it to another molecule. That's what fixing means. Reduction reactions, where we're going to be adding those high-energy electrons. We're going to reduce the molecule. And then we're going to regenerate the molecule we started with that we originally fixed the carbon to, and you'll see that shortly. Okay, so here's stage 1, the carbon fixation stage. This is called C3 photosynthesis because the molecule that I'm going to make has 3 carbons in it. That's the key here. We are also going to talk about C4 photosynthesis after we've got this solid. So C3 is your normal photosynthesis. Every plant can do this. This is your goal here. And what we're going to end up doing is taking our carbon dioxide, you can see it free here, and we're going to add it to this molecule down here, which is called ribulose 1,5-bisphosphate. We call it rubip for short. Ribulose 1,5-bisphosphate. What we have not shown you is all the little functional groups sticking off of this molecule. So when you're looking down here and you see these black grayish things, these are meant to represent the carbons. So it's a 5-carbon molecule. That's the ribo part. Remember, ribose is also a 5-carbon sugar that's in our DNA and our, in our RNA. That's the 5-carbon sugar versus glucose, which is a 6-carbon sugar. So I'm taking another version of a 5-carbon sugar, and notice it has a phosphate on this end and a phosphate on that end. So we say it is a bisphosphate, not biphosphate, but bisphosphate is how we talk about having those 2 phosphates. Okay, bisphosphate. And we call it 1,5 because there's a phosphate on carbon 1 and there's a phosphate on carbon 5. So it's just named after where we place things. So it is a ribulose sugar, but on 1 and on 5, I've got phosphates. Done. That's the name of it. Just call it rubip. Okay, rubip is the full name of the sugar, or I guess I should say here rubip. Rubip is the name of the sugar. The name of the enzyme that's going to grab the CO2, so grab CO2 here and grab this the rubip, is rubisco. Rubisco is a shortened form of this. What we actually tend to shorten a lot of things because these are big, long, horrible words. So you'd actually call it Ru, which stands for, well, you'll see in a second. Ru, bis, co. It's actually 3 words shoved in together. And what it really is rubip, so you can see it up at the top here, rubip carboxylase. That's its full name. What the heck is a carboxylase? Well, ASE tells me it's an enzyme. What is it an enzyme doing? It's adding a carboxyl group. Well, what's a carboxyl group? That's a carboxyl group. Isn't it? It happens to be missing the H on the end, but the oxygens were always the important things anyway. So that's my carboxyl group. What does that molecule look like to you? Count up the atoms. Isn't that just a C with 2 O's? Isn't that just a carbon dioxide? So if I have a carboxylase, it's the enzyme that adds a carboxyl group, which is basically carbon dioxide. Who does it add it to? It adds it to rubip. That's how you name this sucker. So it is the enzyme that adds a carboxyl group to rubip, but for short we call it rubisco. Or you don't have to do capitals, that just helps you to remember it's a 3-part thinger. Rubisco is fine if you do a lowercase, I don't care. Okay, so rubip itself, the actual rubip, and the carbon dioxide get crunched together by rubisco. Notice that there is water involved here, but you're going to see water in and out all over the place with chemical reactions. As far as the overall photosynthetic reaction goes, like the main big one, we really don't care about this water because this exact amount of water will come out, so there's no net gain of water here or net loss of water. Whereas the overall photosynthetic equation, let me show it to you just so it's in your mind. So in this case here, you've got carbon dioxide and you have this water here. And remember that this water just got split through photolysis to make this oxygen. But there's water in on this side and there's water in and out on this side in the different processes. We don't count up any of those other waters because the exact number in and out, they cancel each other out. It's like no net gain or net loss. But here, for sure, I'm losing 6 waters because they are becoming 6 oxygens. So this is just my overall in, out, all over the place. Those things all cancel. It's kind of like if you have an equation and you have X on both sides of the equation, you can cancel them out. In and out doesn't matter. So here, water's all over the place. So do not think about in that other cycle, in this cycle here, that works there. Don't consider this to be an important water. I don't even care if you remember that it's there. The only water I care about is the water we saw at the beginning with photolysis. So again, we have carbon dioxide. Okay, here's our carbon dioxide being fixed to the ribulose 1,5-bisphosphate, which is our rubip for short. And you can call it rubip all day long. Any conversation that you type, just call it rubip. But you do have to recognize if I say ribulose 1,5-bisphosphate, you need to be like, oh, Pereira, that's just rubip. And so that you know what it is, but you never have to write that out. I'm not going to ask you to. I'm not going to check your spelling or anything like that. Okay? If you're brave enough to write it, go for it. But that's probably precious seconds lost. This probably takes 5 minutes or so to write this word. So if you can afford the time on the test, feel free to write it out. But rubip is fine. And then Rubisco, of course, being the enzyme that does the job of crunching those two guys together. Okay? So we used to have our rubip that's already there in the stroma. Carbon dioxide has diffused into the chloroplast, so it's there in the stroma as well. Rubisco is in the stroma, and it gets added together. What does it create? Well, temporarily, the 1 plus the 5 carbons creates this temporary 6-carbon molecule, but it's really, really unstable, mostly because you have these phosphates at either end that are tugging on the molecule. So when I pop this guy into the mix and I add it in, I'm actually creating a very unstable, destabilized molecule that falls apart, and it'll fall apart into these 3-carbon molecules called 3 because of where the phosphate is, because of which carbon the phosphate's on, phospho because it's got a phosphate, glycerate because that's what the carbons are with all their functional groups. Remember, we're not bothering to show all the OHs or Hs or what the double Os and double bonded O's. We're not showing anything that's on the carbons. We're just simplifying this as much as possible. Okay, so we're going to create 3 PGA. So rubip plus carbon dioxide through the magic of Rubisco makes 2 times 3 PGA. And notice you can see that it's doubled up because if you look at this, there's 3 of these and 3 of these, but now there is 6 of these. And if you count the carbons, it would have been 3 times 5 is 15. 3 times 1 is 3. 3 of the carbon dioxide carbons plus 15 of the rubip carbons gives me 18. 18 chopped into 3 gives me 6 of these 3 carbon molecules. And if you followed that, I am very impressed. Okay. That is stage number 1, fix the carbon. And it's a simple conversation. Again, it's carbon dioxide gets fixed to rubip by Rubisco, forming 2 3-PGAs. More nasty words, but they're not bad once you realize what they're talking about. Okay, so let's run through it. First of all, we mentioned endergonic and exergonic when we're talking about enzymes. Are you trying to make a bond so you can store energy? Then you've got endergonic. If you are trying to break a bond so that you can release energy, that's exergonic, right? So in this case here, it's a reduction reaction, which means I'm going to take that high energy electron that I just packaged into the NADPH, and I'm going to deliver it now to these molecules here. And so it's going to be a 2-step process because not only will I use the NADPH, but I'm also going to use the ATP as well. So we end up having ATP being hydrolyzed or going through hydrolysis. What type of reaction is hydrolysis? It's catabolic, which is a fancy way of saying, what am I doing to the ATP? I'm taking it apart. Okay, what are the component parts? It's an ADP and a phosphate, right, that I put together earlier. So I'm going to break off that phosphate, and we mentioned that phosphate, which was shown right here, this bond that's right here that I have the slash through, this is what we're going to cut. That's a high energy bond because it took a lot of energy to just shove those phosphates together because they don't want to be together. So the fact that I got them to glue means that's my gorilla glue. I must have had lots of energy holding them, right? So I've got a high energy bond right here, and I've got the high energy electrons built into this relationship here between NADP and the electrons with the package of a hydrogen that I shoved on the end. Okay? All this energy is going to be released now and added to the molecules to help reshape them. So water is going to do the job through hydrolysis of breaking this bond here between the phosphates. So then I'm going to end up with a DP, diphosphate, and an inorganic phosphate. It's the exact reverse of what we just did 5 seconds ago. In the second section of it, I'm going to take the 3-PGA that I just made. My thing is frozen. Here we go. 3-PGA is this guy right here. The one that just fell apart after I added the carbon dioxide to the rubip. So I'm going to take the 3-PGA. I'm going to take the 3-PGA, I'm going to use that ATP energy after I break it off and the energy from that high energy electron, and I'm going to rearrange the sucker. Now, if you're looking at these drawings here, or even if you're looking down below at the next set of drawings, 3-PGA in this diagram sure looks a heck of a lot like this guy. What's the big difference in these two pictures? So I had one phosphate group at the 3 carbon, that's why it's 3-PGA. All I'm going to do is add another second phosphate, and that's where the ATP comes in. I'm going to take the phosphate right off the ATP to phosphorylate this glycerate molecule. Glycerate tells you all the stuff that's sticking off of the carbons that we haven't bothered to draw. So a glycerate is a 3-carbon molecule with a bunch of stuff around, and then it has a phosphate, so it's a phosphoglycerate. This one also is the exact same stuff on the carbons, but now it has 2, so it's a 1,3-bis phosphoglycerate, because that's how we say bi. We don't say bi, we say bis. And that's it. So we go from 3-PGA to 1,3-BPG. We're actually going to see 3-PGA and 1,3-BPG again, because these molecules head us towards glucose. So it would make sense that if I began to break glucose down, I'd have to go through these intermediate molecules to get me back to just having free carbon dioxide. So I'm actually going to be using these again in our next conversation. You'll see them in like glycolysis lesson. Okay, don't have to remember that right now, but you will see them come back. So I used the ATP energy that I created through chemiosmosis. I stole that phosphate off it, gave it to the 1,3-BPG. Why the heck would I ever want to do that? Well, what it's going to end up allowing me to do, along with the high energy electron that's coming from the NADPH, it's going to allow me to rearrange it from being a glycerate, remember we're talking about the stuff that's attached to these carbons, to a glyceraldehyde. That's the difference. It's the functional groups are going to get rearranged, and I'm going to use the energy from that high energy electron, originally that came from the chlorophyll, from the ATP, use that energy to rearrange a molecule. Now this molecule, the G3P version, is a half sugar. It's ready to become glucose. That's all I did. Grab some energy, use that energy to rearrange a molecule that's similar but not identical. Now that I've got the functional groups rearranged, I'm ready to make sugar. That's it. That's how light becomes sugar. That's the entire photosynthesis conversation in terms of, like, if we're just doing it very generally. So does the light actually become sugar? No, the light provides the energy that allows us to shift an electron that then allows us to shift functional groups on a molecule. And again, we're not showing all the functional groups, so you can take it as it is, but this is what it is here. Okay. There's one more thing that we need to do, though, because if we go back up to the top here, when we first fixed carbon, okay, carbon fixation, we had added it to this molecule here. If I run out of rubip, none of this can happen anymore. I have to have enough rubip to make the whole process work, right? So what I'm going to do is, my first stage, fix the carbon. Second stage, use my energy to reduce, add some electrons, move some things around so I can have my G3P, or PGAL. And then I have to make sure that I also make some more rubip so I can start again. And that's my third step, just make more rubip. It's called the regeneration of rubip. Sounds very fancy, but it just means make more. I do want to mention, before I move off of this page again, I've said this before, but I'll say it to you again. There are 2 names for the main thing that we're making here, for that half sugar that we care about. We can call it PGAL, or we can call it G3P. The difference in the name is whether you're being very specific or not. Notice it's a glyceraldehyde. That's the main carbon structure of it. Okay? Glyceraldehyde is in both names. Glyceraldehyde, glyceraldehyde. They're both phosphos because they have a phosphate attached. So it's either phosphoglyceraldehyde, PGAL, or glyceraldehyde 3-phosphate. Why put the 3 in there? Why is that important? It tells you what carbon the phosphate is attached to. Happens to be on carbon 3. We always name the carbons in a ring or carbons in a chain so we can tell what's happening where. So carbon 3 has the phosphate. So G3P is just a more specific name than PGAL. You can use either one. And again, you never have to spell them out for yourself, but you do need to recognize them in case I use them in a matching question or something. And you do need to know that these are the exact same molecule. There'll be a few of them that they'll have two names, and you'll just have to make sure you know which ones belong with which names. That actually sometimes even becomes little matching questions or little multiple choice type of questions. Okay, so what's the whole goal of the second stage? It's to use the energy in the ATP and the NADPH to help us rearrange a molecule, fix the carbon, rearrange a molecule to get me to this G3P stuff. Because G3P is a half sugar that I can then use to make glucose or any other sugar that I want. So one more time, second stage, use the energy from the ATP, use the energy from that high energy electron to rearrange from 3-PGA, so 3-phosphoglycerate, to a 1,3-bisphosphoglycerate, and essentially ending up at a glyceraldehyde or G3P or PGAL. You'll probably end up like me where it's interchangeable. Sometimes you say G3P, sometimes you'll say PGAL, and you won't even realize that you're switching between them. So what are we going to do with this? Well, one G3P goes off to make sugar. So we call that a half sugar. So in other words, I have to run this twice to get a full sugar. The other 5 that I've produced, because I make 6 of them, you see there's a big 6 times over there. One of them goes off, becomes a half sugar. 5 of them I'm going to use to make more of this rubip that I started with so I can keep this cycle going. I hope you can recognize what you're copying here is the little darker purple dots are my carbons, and those are the phosphates. I've just tried to replicate the way they've already been drawn. So what are you looking at here? It's our final stage. We're taking 5 of the G3Ps that we made. We made 6 total, but 5 of them here. And I don't need you to memorize this at all, but you can see there is more ATP involved. But what we're going to do is rearrange, 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 rearrange. There are intermediate molecules here that we're not bothering to show. And I should also mention, this doesn't just magically happen. For every arrow that you see, there's at least one enzyme doing this job. So you have machinery in there, protein machines that are making all of this happen. So you probably thought, clearly these represent magic. Unfortunately, they do not. They represent chemical reactions that are happening, forced by these catalysts, these protein enzyme catalysts. Okay. You don't need to remember this. What you need to remember is that we take some of the G3P and we make new ribulose 1,5-bisphosphate so that I can start the whole cycle over again. Okay. So what's all this little junk here about? Well, we've got 5 of these, and if you count it up, it's 5 times the 1 phosphate, so I've got 5 phosphates. Then I've got 5 times 1, 2, 3, so I've got 15 carbons total in my pool. Then I'm going to have to add in a few more of these phosphate groups. Notice I've actually knocked off a couple phosphates, so 2 came off, but 3 came on. And the 2 came off simply because I had to rearrange the molecule somehow. Just in between, we're not showing the intermediates, right? So in the process, I lost a couple of the phosphates that were attached to join some stuff together. And now I'm going to add in some more so I can get the other ones on the other ends of these new molecules that I'm making. That's it. And then, so now when I count them up, I can say I've got 3 of these big rubips. So 3 times the 2 phosphates on either end give me 6 phosphates. 3 times the 5 carbons in the middle give me 15 carbons. Okay. Then I have these 3 ADPs, and I have the 2 inorganic phosphates. So if I added them all up, you'd see on either side of this equation, we have the same number of phosphates and same number of carbons in and out, no problem. Okay. Again, what you need to remember is that I have to regenerate rubip. That's your conversation. G3P, most of the G3P is used to regenerate rubip. Stop there. You don't have to memorize how many they are. It might be helpful to remember it's a 5-carbon sugar, but it's really not the end of the world if you forget that, as long as you remember what rubip is for. So again, to make that glucose, you're going to need 2 of those G3Ps, or 2 PGALs. So this whole thing has to run twice, because each time 5 of the 6 are going to be needed to go this way. So I've only got 1 of these 6 G3Ps to go down to make sugars. So it runs twice to get me the extra 1 to make the full sugar. Now, depending on what the plant needs will depend on exactly which sugar I produce. And if I am producing glucose or fructose or whatever, then I need to think about how do I transport it to the rest of the molecule, and then how do I store it, or how do I use it for structure like cellulose. Right? So this is the next conversation. So because G3P is already a 3-carbon sugar, and glucose is a 6-carbon sugar, I can use it directly to make glucose. If I want to transport it though, why transport 1 glucose at a time when you can transport 2 sugars at a time? So this is glucose and this is fructose, right? That's what we talked about sucrose at the beginning. I said you wouldn't have to draw it, but it'd be good to kind of tuck it away and memorize. So if I put a glucose and a fructose together, remember as a disaccharide, it's still soluble in water. I can still dissolve it, which is great because then I can throw it into the phloem tube and I can transport it really easily as this double sugar. Awesome. Just based on its own properties, right? So glucose then is my main sugar. I'm making it from 2 p-gals. Sucrose is how I'm going to try to transport it, and this will happen out in the cytoplasm. It doesn't have to happen in the chloroplast. This happens in the chloroplast possibly. Could be in the chloroplast, could be out in the cytoplasm, just depends. And I'm going to transport it either to any plant cell that needs sugar for its energy. It's going to use it for cellular respiration, right? Or go store it somewhere. You can go use the phloem tubes to store it in the roots, store it in the stem, wherever your storage happens to be for that type of plant. In addition, once the sugar gets there to these storage cells, I take a whole bunch of these and I just begin to click them in together. I keep adding these little monosaccharides and I get longer and longer and longer and longer chains of sugar. In this particular case, we have starch, and this little box up here is meant to represent the CH2OH group, right? Notice that in starch, they're all sticking up in the same direction. This is forming an amylose sugar. And if I want to chop it back down again, I'm going to have to chop it down with an amylase enzyme. So you have an enzyme that'll build it, and you have an enzyme that'll break it down. If I wanted to like chop off little sugars at a time. Finally, the last type of sugar that plants have is cellulose. Please tell me that you can see the difference between a cellulose and a starch. What's the major difference here? The CH2OH is up here and up here in the first sugar, but here it's still up and over here it is down. So we have an alternating, you're actually flipping the sugar up and down every other. You can, in your body, when you eat plants, you can break down starch. No problem, because your enzyme is able to get in there and it actually breaks this exact bond right here. Because if I can chop off 2 of these at a time, I can make maltose. Remember maltose is that double sugar you guys drew? So I can come along and I can physically, as an enzyme, I can come in here. Actually, I'm probably coming in here. I can latch onto the last 4 and I can cut right in the middle. But if I try to latch on here and chop those last 4 in the middle, these guys here that are pointing down block me from being able to fit that into my active site. So this substrate will not fit in the amylase enzyme. I can't do it. I need a different enzyme called cellulase that can break down cellulose. This is why when you eat lettuce or whatever, any type of this greenery, you cannot break down cellulose. You don't have the enzyme available to do that. This is why it becomes fiber in your diet. Still valuable in your diet, but it doesn't provide any energy or any nutritional value of actually being digested and sucked into your body to be used for parts. It's just there for bulk. It remains, I'm going to use the word lumen, it remains in the lumen of your intestines, the inside of your intestines, and just helps to push the bulk through. And it's all because of the way that these little functional groups are arranged, the way that these sugars are upside down and they alternate. Let's look just at the light-dependent part. So the first thing that we did in that lesson, and we're talking about what we needed. First of all, we need to talk about where were we? We were in the thylakoid membrane, right? So the light-dependent happens in the thylakoid. Why? Because that's where the machines are anchored. So we've got our chlorophyll anchored in the photosystems. We've got these electron carriers anchored. We got the NADP reductase anchored. And we can, that Z protein can split the water on the inside. Great. Everything is in the right spot, nice and tight and close together, so that this is a very efficient process. I don't have to worry about my electrons going the wrong direction. They will go where I want them to go. Okay. What did we put in? We put in water, and then we put in NADP and those protons. And of course, they were glued together by the electrons. What did they become? Oh, sorry. And then we also put in ADP and the inorganic phosphate. What does the water become? The water becomes these 3 parts. Then what does the NADP become with that proton? It becomes NADPH, and ADP becomes ATP. It's not very difficult, though, if you remember the three important things. You've now hopefully locked down the idea of photolysis. So if you know water is put in, then it should be easy to remember the three parts water splits into, isn't it? And you should know by now that the whole point of this entire process was to produce the NADPH and the ATP. How did you get that? Well, you should know what the component parts are. ATP is ADP and an inorganic phosphate. NADP plus the proton give me the NADPH. So if you just remember those three important things in and out, you should be able to get the reverse of them split apart is much easier. Again, we're always compartmentalizing as you're memorizing so that you know piece by piece. What's great about it is it also makes the next part very easy as well if you compartmentalize. So when we're talking about the half reaction, you can see that we have water in. All that other junk is just going to get us to the energy molecules that we care about over here. And you can see that we have oxygen out. The reason I boxed the water and the oxygen is because they belong in the overall equation for photosynthesis. Those are the important components there. But the fact that we have NADP in and ADP in doesn't matter because in the next half of the reaction, I'm going to get those out again. So overall, they're going to end up canceling. You'll see that in a second. So now we see where does it happen? In the stroma. I'm out in the jelly. Still in the chloroplast, but in the stroma of the chloroplast. What does it require? This time, I'm going to put in CO2, but I'm going to get out G3P. I'm also going to put in rubip, but I get out rubip. So that's a nice, easy one to cancel out. And same thing again, I'm going to use the ATP that I created earlier, and I get out those two parts. I use the NADPH I got out earlier, and I get out those two parts. So again, all we're doing with this half reaction is just writing what's in and what's out. Notice that carbon dioxide comes in. How do you know that's part of our main equation? What's the goal of putting in the carbon dioxide? I want to get out this G3P, and of course the G3P is just there to help us make glucose. So there's the other important part of our overall equation. I had rubip in, but I had rubip out. So that cancels. It's on both sides. I had ATP in and NADPH in, and then I get out this stuff on the other side. Okay. But when I combine the two half reactions, I started with this, and then I end with this junk. I created this junk, but I used it. So that all cancels out. So literally the only thing that ends up being left are the things that I boxed out. Those are the important things. So the water and carbon dioxide in, because you never see those guys again, and it's oxygen and G3P out, and of course we're skipping the stage where we tell you how the G3P becomes glucose. So we're just going to treat it like as if that stands in place of our glucose for our overall equation. So then when we do look at the overall equation, water in because of photolysis, carbon dioxide in during the carbon fixation stage of the Calvin cycle. A photon of light helped me to energize those electrons. I ended splitting the water, and so that I produced that oxygen, and my end goal of fixing the carbon was to make finally this sugar molecule, probably a glucose. That is photosynthesis. It's a lot of steps, but if you spend a little bit of time with it, it's really not that bad. I hope you get to that point where you don't feel like that. And hopefully that point is before the test, because that would be the most convenient time to know what you're doing. So here's the big picture all in one. So we have 3 stages. Number 1, fix the carbon. What's involved? Rubip plus carbon dioxide gives me some 3-PGA. 2 times 3-PGA. What do I do with that? I take my 3-PGA and I rearrange it to G3P. Again, it's the difference between a phosphoglycerate and a glyceraldehyde. Glycerate versus glyceraldehyde. Okay. I use one of them to go off and make a half sugar, and I use 5 of them to go off and make some more rhubarb. So that comes back around to rhubarb. So I have stage 1, fix the carbon. Stage 2, use the ATP and the NADPH in the reduction reactions. Stage 3, make more rhubarb. And that's literally all I need to know first for stage 3. Makes more rhubarb. Just for interest's sake, there's no need for you to memorize this diagram whatsoever. It's just showing you where the actual carbons are and the fact that we do have things sticking off the carbon that we just didn't comment on. So you've got the black is meant to represent the carbons. You've got the red representing the oxygens. The little white ones are your hydrogens. And then the purpley colored are the phosphorus atoms, which of course are arranged in phosphate groups. So we have the rhubarb at the top, fixed to the carbon dioxide, or I should say the carbon dioxide is fixed to the rhubarb. Then we rearrange. Notice that we've made 2 of these guys, 2 of the 3 PGAs. Then we use our ATP to add a phosphate. We make 1,3-BPG. Use the NADPH to destabilize and rearrange some stuff. So it's the carbons, the stuff hanging off the carbon that gets moved around a little bit, knocks off that phosphate, change it, end up with ribulose 5-phosphate, ends up with, or 1,5, I guess, bisphosphate at the end. That's it, done. Okay, fix the carbon, rearrange the molecule. Some of them go off to make sugars, some of them go off to make more rhubarb. I'm done. That, my friends, is the end of the conversation for C3 photosynthesis. What I want to do right now is give you guys just a few moments to kind of absorb that, talk it out, walk through the cycle, make sure you understand the 3 little mini stages. Say the word rhubarb, say carbon dioxide, say rubisco is the enzyme that fixes the carbon dioxide. That's the new term, fix the carbon dioxide. Talk about it rearranging. So 3-PGA, 1,3-BPG. You can even do a little rhyming thing. Just give yourself a few minutes to absorb that because I'm about to add to the conversation and I don't want to confuse you.